Hello, my name is Devleena Samanta. I'm a postdoctoral fellow in Professor Chad Mirkin's lab at Northwestern University. In this biomimetic materials session, I'm very excited to be sharing with you some of our recent work on how we can use DNA to build dynamic materials based on bio-inspired approaches. Now, you probably know of DNA as being the building block of life. But in this talk today, I want to introduce to you the concept of using DNA as the building block of materials. So this figure shows the evolution of DNA-based materials over the past 25 years or so. But this figure is rather involved. If I wanted to simplify this figure, what I could do is I could say that the advances in this field could be broadly categorized into two main groups. First, DNA can be folded just like origami to form its own structures. And we can really form structures of arbitrary complexity. For example, we can make a smiley face that's made entirely out of DNA. On the other hand, DNA can be used as a bonding element to direct the organization of nanoparticles into hierarchical structures. While these two ideas are somewhat related, there's an important distinction in the second case. Specifically, in the second case, we can use abiological structures, something like functional nanoparticles, and arrange them in three-dimensional space with a degree of structural control and precision that is not afforded by any other technique. So this idea was pioneered by the Mirkin group, and it has grown to be a very important concept in material science for building materials from the bottom up. So at this point, there's an important question that comes up, which is what is so special about DNA that makes it so powerful that has created an entirely new field in material science? Well, to answer this question, we need to really look at the structure of DNA. So here I've shown the quintessential double helix cartoon structure of DNA. But if we were to zoom in to the structure of a single strand, what we see is that the DNA has a sugar phosphate backbone, which is negatively charged, and some bases. And these bases, we know, have very specific interactions. So we know that A binds with T and G binds with C. And as a result of these specific interactions, if we were to have two different single strands of DNA that have complementary bases, they would interact with each other and hybridize to give you the double helix structure that's shown on the left. So this tells us that DNA is a very programmable molecule. Beyond programmability, DNA also has some very advantageous properties, as a result of which it has emerged as a really attractive material building block. And if I had to sum up all these advantages into a single sentence, what I can say is that DNA can be used to build very modular structures with designer sequences of any given length relatively quickly and relatively easily. Now, when we think about building materials, the first thing that comes to mind is the periodic table. And this is because the periodic table provides a menu of every single element that is known in the universe. And all materials are nothing but a combination of one or more of these elements. But the thing about atomic systems is that their valency is more or less immutable. And what that means is that we can't really force any element to adopt any valency. Now, the valency of elements determines their bonding characteristics, which in turn determines the types of structures that are possible when these elements interact with one another. So imagine an artificial system, let's call it an atom equivalent, except in this case, we can decouple the identity of the atom from its bonding characteristics. So this would imply that we can use any material building block and position it in three-dimensional space in any way we wanted. So this would be a really powerful material building block. So this is possible by using a DNA functionalized nanoparticle as the building block. So here, these constructs are referred to as programmable atom equivalents. The nanoparticle core can be thought of as an atom, and the DNA shell provides the bonding characteristics. So these particles are densely functionalized with DNA that terminate in a single-stranded region, which we call the sticky ends. So if we were to have two sets of nanoparticles, that have complementary DNA, then these two sets of nanoparticles would interact with each other and form DNA bonds between them. And the same way atoms come together to form crystals, these colloidal particles can come together to form colloidal crystals. And we can even visualize these crystals by electron microscopy. So here in this example, a crystal that is made out of gold nanoparticles is shown. The reason that this strategy is really powerful is because by changing the shape, size, and composition of the nanoparticles, as well as by changing the DNA design, we can have access to a library of different structures. Till date, over 500 different structures have been achieved, but the caveat here is that most of these structures are static. So the question that I wanted to ask is, how can we make structures that are dynamic? Dynamic materials are important because this would indicate that we would have to synthesize a single material and we would be able to control or change its properties by changing the interparticle distances by using some sort of stimulus. So we can start to make things like flat lenses or smart windows. So this strategy would really give us access to dynamic optical, catalytic, magnetic, or even mechanical metamaterials by using appropriate nanoparticle building blocks. So how can we build dynamic materials with DNA? To do this, we looked at nature for inspiration. In nature, DNA exists in a highly condensed state. So if we were to take out all the DNA in a single living cell and stretch it out, that would be about 6 feet long. But all of this DNA exists inside the nucleus of a cell that's roughly 6 microns in diameter. How does this work? Well, we can think of it like a ball of wool. In case of DNA specifically, we know that the backbone is negatively charged, and this DNA interacts with proteins that are called histones, and these histone proteins have multiple amino acid residues that are positively charged. So as a result of the interaction with these multiple positive charges, the DNA exists in a highly condensed state. So this then led us to hypothesize that if we were to synthesize DNA-mediated colloidal crystals and add cations that have multiple positive charges, these multivalent cations would probably induce a condensed state of the DNA, which would then lead to a global structural change and lead to a global contraction of the superlattice. And if we were to remove these cations, perhaps we can expand these crystals back. This work was highlighted on the cover of JACS earlier this year. To test our hypothesis, we used two sets of nanoparticles that are gold nanoparticles, and these nanoparticles were of identical size and DNA length, but they had complementary DNA. So in this case, we would expect a body-centered cubic lattice, which we could confirm by small angle X-ray scattering studies. And this type of body-centered cubic lattice would be expected to form a rhombic dodecahedron crystal habit, which we also confirmed by electron microscopy. Now that we have synthesized our colloidal crystals, next we wanted to test our original hypothesis, which is what happens when we add increasing concentrations of multivalent cations. So we started by adding nickel, and here from the SACS data, what we observe is as we add increasing amounts of nickel, we see a right shift in the Q values. So this would indicate that the inter-nanoparticle distances are becoming smaller with increase in concentration of nickel. We also observe as we gradually decrease the nickel concentration back, we see that the peaks revert back to their initial positions. From this data, we can calculate the surface-to-surface distance between the nanoparticles, which is what we refer to as the DNA bond length. And then we plot this DNA bond length versus increasing concentrations of nickel. So what we observe from this data is that the DNA undergoes a significant change in length. So in our experiments, we found that we can obtain up to 85% in change in the length of the DNA and up to 80% change in the volume of the crystal. And these changes are completely reversible. So when we remove these metal ions, we see that the crystals go back to their original state. We could also observe that the crystallinity is retained throughout this process, and we could observe that both through our SACS data as well as through imaging by electron microscopy. So this is really impressive that we are obtaining such large deformations, yet the order and crystal habit are being retained. We next wanted to investigate how the charge of the cation influences the extent of contraction. And what we observed is cations with greater positive charge induce more contraction. However, charge alone cannot explain the extent of contraction that is observed. For example, if we were to look at magnesium and nickel, both of which are divalent, we see a significantly more contraction with nickel than with magnesium. This is more apparent when we do concentration-dependent studies, where we find that a 2-molar salt, magnesium only induces about 30% contraction, whereas nickel induces up to 80% contraction. Now, even though we could study the global changes in structure by using SACS and electron microscopy, we couldn't really look at the structural changes in DNA using experimental methods. This is why we turn to molecular dynamics simulations. And these images correspond to the molecular dynamics simulation generated structure of DNA at the experimental interparticle distances when 2-molar salt is used. So what we observe here is that when a high concentration of nickel is used, the DNA adopts a very, very twisted and bent structure. And what we also learned from molecular dynamics simulations is that even though we add magnesium and nickel at identical concentrations, the local concentration of nickel around the DNA is significantly higher, indicating that the binding affinity of these metals to DNA is an important dictator of the extent of contraction. So to test this hypothesis, we looked at different metals with different binding affinities, and what we found is indeed metals with higher binding affinities to DNA induce a greater extent of contraction. When we think about dynamic systems, we not only think about what is the extent of actuation, but we also think about how fast are these changes occurring. So in most micron scale systems, the time frame is of the order of minutes to sometimes even hours because of diffusion limitations. In this case, we observed the contraction process in real time as we added 2-molar nickel. And what we observed is that actuation occurs on sub-second timescales. On the right, you can see what is happening to the DNA structure based on molecular dynamics simulations. And it is really impressive that despite the large deformations, we see no loss in DNA base pairing. To summarize, inspired by histone proteins that condense DNA in the nuclei of living cells, we have developed a strategy that allows one to reversibly contract and expand DNA-mediated colloidal crystals. This strategy relies on the interaction of multivalent cations with DNA and allows one to tune interparticle spacings with sub-nanometric precision. We also learned that cations that bind more strongly to DNA induce greater contraction. What is really impressive is that in this process, we can get up to 85% change in the length of the DNA, which can bring the particles within 3 nanometers of each other, which is important for plasmonic or magnetic coupling. And this corresponds to about 80% change in the volume of the crystal. And despite these large deformations, the lattice symmetry and crystal habit are retained throughout the process, and the actuation occurs on sub-second timescales. So in a nutshell, this strategy provides access to a new class of optical, catalytic, magnetic, and even mechanical metamaterials. In this talk, I told you about how we can use DNA functionalized nanoparticles as programmable atom equivalents for materials applications. However, these structures also have many emergent properties, which has tremendous biological implications. In the context of biology, we refer to these structures as spherical nucleic acids, and one application of these in the biological context is to be able to look at analytes in live cells. If you want to know more about my work in that area, please view my second talk. I would like to thank Professor Mirkin for providing me the opportunity of working in his lab And I would also like to acknowledge all of my wonderful collaborators for making this work possible. If you have any questions, feel free to contact me either through the AIChE chat or via email. Thank you for your attention.